A new scientific paper examining a genetic material recovered from the Shroud of Turin was published in early 2026, and within days, it had spread across religious news outlets, science blogs, and social media, generating exactly the kind of intense reaction that anything touching this particular cloth tends to produce. Vatican News covered it. Smithsonian Magazine covered it. Dozens of smaller outlets picked it up and ran with increasingly dramatic headlines. But underneath the excitement sits a much more interesting and considerably more nuanced story than most of the viral coverage actually captured. A story about what modern genetic science can and cannot tell us about an object that has been touched, handled, displayed, transported, and venerated by countless people across nearly 7 centuries of documented history. To understand why this study matters, and equally why its actual findings are more complicated than a lot of the coverage suggested, it helps to go back to where the genetic material being studied actually came from in the first place. In 1978, during one of the most extensive scientific investigations ever conducted on the shroud, a researcher named Pierluigi Bologna collected physical samples directly from the cloth, which is permanently housed at the Cathedral of St. John the Baptist in Turin, Italy. Those samples sat preserved for decades, waiting for genetic sequencing technology to advance to the point where they could be meaningfully analyzed. Bologna remained involved in shroud research for the rest of his life and was part of the team behind this most recent DNA study before passing away in late 2025, meaning this new research represents something close to the final chapter of a project he helped begin nearly 50 years earlier. The actual study, formally titled as an analysis of DNA traces from that 1978 sample collection, was led by geneticists, including Dr. Gianni Barcaccia of the University of Padua, and it used a technique called metagenomics, essentially a method of sequencing and identifying every distinguishable genetic signature present in a sample, rather than searching for one specific target genome. This distinction matters enormously for understanding what the studies actually found, because metagenomic analysis isn't designed to isolate and confirm the DNA of one specific historical individual. It's designed to catalog the full, messy diversity of genetic material present in a sample, which, for an object like the shroud, means centuries of accumulated biological residue from everyone and everything that ever came into contact with it. And that's exactly what the researchers found: a genuinely striking diversity of genetic material that the study's authors themselves described as a diverse mosaic of genetic traces. The human DNA recovered showed signatures connecting to multiple distinct geographic regions across several different centuries, including genetic markers, associated with the broader Mediterranean world, the Middle East, and even more surprisingly, genetic traces connected to India and North America. Vatican News, reporting on the findings directly, noted that the researchers confirmed the presence of a genome predominantly linked to the Middle East, along with detecting microorganisms known to thrive specifically in extremely saline environments, the kind of environment associated with a region like the Dead Sea. That particular detail is genuinely interesting, since it aligns with historical traditions placing the shroud in the Middle East at some point during its history, though it's worth being careful about exactly what this proves. Microorganisms adapted to saline. Environments could theoretically end up on a cloth through numerous pathways across centuries of travel and handling, and their presence indicates exposure to that kind of environment at some point, rather than confirming precisely when that exposure occurred or definitively proving the cloth originated there in the 1st century specifically. Beyond the human genetic material, the study also identified an extensive and, frankly, somewhat surprising range of animal DNA present on the cloth. Researchers found genetic traces from cats, dogs, chickens, cows, goats, sheep, pigs, horses, deer, and rabbits, along with traces from various fish species, including gray mullet and Atlantic cod, marine crustaceans, flies, aphids, and even arachnids. That eclectic collection makes considerably more sense once you consider the shroud's actual documented history, which involves centuries of transport across different regions, storage in various locations, direct handling by countless individuals, public displays before large crowds, and even survival through a serious fire in the 16th century. A physically eventful history for any object, let alone one made of delicate ancient linen. Every single one of those events represents an opportunity for biological material from animals, insects, food, textiles, and human handlers to become embedded in the fibers. This is exactly why the researchers themselves were notably cautious in how they framed their own findings, a level of caution that got somewhat lost in the more sensationalized headlines the study generated. According to the researchers, DNA testing could not definitively date the shroud, and rather than confirming any single origin story, the results instead illustrated just how extensively the cloth has been handled by different people across many centuries. That's a genuinely important finding in its own right, since it adds real hard genetic evidence to something historians already suspected based on the shroud's well-documented history of public veneration and transport. But it's a meaningfully different claim than decoding Jesus' blood or delivering some kind of definitive scientific verdict on the crucifixion itself, however dramatic that framing sounds in a headline. It's worth pausing here to separate this DNA study clearly from a different piece of shroud research that sometimes gets blended together with it in less careful coverage. A study conducted by Giulio Fanti, a professor of mechanical and thermal measurements at the University of Padua, focused specifically on the physical pattern of bloodstains visible on the cloth rather than genetic material. Fanti's analysis examined the macroscopic and microscopic behavior of the bloodstains themselves, arguing that the way blood flowed on the shroud, showing 3 distinct directional patterns, a vertical flow consistent with an upright body position, an inclined flow at roughly a 45-degree angle, and a horizontal flow consistent with the body later resting on its side, matched the physical mechanics of what would be expected from a crucifixion victim's body being moved through those specific positions. This is a genuinely different kind of evidence than genetic sequencing. It's about fluid dynamics and wound physiology rather than DNA identification, and it represents a separate research thread that happened to generate headlines around a similar time as the metagenomic DNA study, leading some coverage to blend the two together as though they were one unified discovery, when they're actually distinct pieces of research using entirely different scientific methods and answering different questions. There's also a third thread worth mentioning because it frequently gets tangled into shroud coverage as well, the question of how the image itself formed on the cloth, which remains genuinely unresolved regardless of what the DNA study or the blood pattern study concluded. Some researchers examining the image at extremely high resolution have noted that bloodstains appear to sit intact beneath the image layer itself, meaning the image did not form in the areas already covered by blood, suggesting the blood was already present on the cloth before whatever process created the visible image occurred. If accurate, that sequencing detail is genuinely interesting from a forensic standpoint, since it suggests 2 separate physical events happening in a specific order rather than a single artistic process applied all at once. But it's important to note that the underlying mechanism behind the image formation itself remains just as unexplained today as it's been for over a century of dedicated research, and neither the recent DNA study nor the blood pattern analysis actually resolves that particular mystery, despite headlines sometimes implying that the crucifixion has now been comprehensively decoded. Now let's address directly the question that any reasonable, curious person should be asking after hearing all of this. Does any of this new research move the needle on whether the shroud is authentic? The honest answer is considerably more complicated than either enthusiastic believers or dismissive skeptics might prefer. The DNA metagenomic study doesn't provide dating evidence at all by the researcher's own explicit statement, so it neither supports nor contradicts the controversial 1988 radiocarbon results that dated the cloth to the medieval period. What it does provide is a detailed genetic snapshot of the cloth's handling history, which turns out to be exactly as extensive and geographically varied as centuries of documented transport, display, and veneration would predict. The presence of Middle Eastern genetic signatures and salt-loving microorganisms adds an intriguing data point that aligns with traditions, placing the shroud in that region at some point. But it doesn't function as independent proof of first-century origin, since contact with that environment could theoretically have occurred at various points throughout the shroud's long and eventful documented history. The radiocarbon dating controversy itself remains exactly as unresolved as it's been for decades, and this new DNA research doesn't change that underlying disagreement in any direct way. The 1988 testing, conducted by 3 separate laboratories, dated the linen to somewhere between 1260 and 1390, and this result remains the dominant reference point within mainstream scientific literature on the subject. Researchers who dispute that dating have continued to argue that the specific sample tested may have come from a section of the cloth repaired following the documented 16th century fire, meaning it could have included newer threads blended with older original fibers, a theory sometimes referred to informally as the reweaving hypothesis. That objection remains, as it has for years, a genuinely contested point within shroud research, rather than something either side has fully settled, and the recent DNA study neither confirms nor resolves this particular dispute, since analyzing genetic diversity is a fundamentally different scientific task than determining the physical age of the linen fibers themselves. It's also worth being clear about a technical limitation that any honest discussion of this research needs to include: the simple fact that DNA degrades significantly over time, breaking down into increasingly fragmented and difficult to interpret pieces the older it gets. This means that even if some portion of the genetic material recovered from the shroud genuinely dates back to antiquity, It would likely be so degraded and fragmented that reliably distinguishing genuinely ancient DNA from more recent contamination becomes extraordinarily difficult using current techniques. In practice, what researchers can most confidently detect and interpret tends to be whatever genetic material is most recent and most abundant, precisely because it survives in better condition and larger quantity than older, more degraded traces. That technical reality is a big part of why the study's authors were careful to frame their findings around the diversity and complexity of the cloth's handling history rather than making any claim about identifying ancient first-century genetic material specifically, let alone genetic material belonging to any particular historical individual. This is really the heart of why headlines claiming a DNA study has decoded Jesus' blood or shocked the Vatican significantly overstate what the actual published research demonstrates. No credible geneticist working on this study claimed to have identified genetic material definitively belonging to Jesus of Nazareth specifically, and doing so would be essentially impossible using current technology and available reference material, regardless of whose DNA might theoretically be present in the sample. There's no verified reference genome from a first-century individual to compare anything against, and even under ideal preservation conditions, which the Shroud's eventful and well-handled history doesn't remotely provide, distinguishing one specific person's genetic signature from nearly 2,000 years of subsequent human contact would be an enormous technical challenge that current genetic science genuinely cannot claim to have solved. As for the claim that this research shocked the Vatican specifically, that framing also deserves some scrutiny. The Vatican has never issued an official declaration on the Shroud's authenticity one way or another, and church leadership has consistently emphasized that the object functions primarily as a devotional aid for reflecting on the Crucifixion, regardless of the ongoing scientific debate about its physical origins. Vatican News did report on the DNA study's findings, which is a fairly routine act of religious media coverage rather than evidence of institutional shock or alarm. Recent popes, including previous public comments on the subject, have generally described the Shroud in terms of spiritual significance rather than staking the Church's credibility on a specific scientific verdict about its age or origin, an approach that has allowed the institution to remain largely unaffected, regardless of which direction ongoing scientific research happens to point. So what should someone genuinely curious about this topic actually take away from all of this new research once the sensational headlines get set aside in favor of what the underlying studies actually demonstrated? The DNA metagenomic research provides a genuinely fascinating and scientifically rigorous look at just how extensively this particular object has traveled and been handled across centuries, revealing a genetic mosaic that includes traces from multiple continents and dozens of different animal species, exactly the kind of complex signature you'd expect from an object with the Shroud's well-documented eventful history, regardless of when or where it originally came from. The separate blood pattern research adds an intriguing forensic data point about the physical mechanics of the bloodstains themselves, worth taking seriously as one piece of a much larger evidentiary puzzle. And the underlying foundational questions, when the cloth actually originated and how its central image was actually formed, remain exactly as contested and genuinely unresolved as they've been for decades, since neither of these newer studies was designed to answer either question definitively, regardless of how the coverage surrounding them got framed for maximum attention. That's not a disappointing conclusion, even though it lacks the dramatic finality a viral headline promises. If anything, it's a genuinely useful reminder of how real scientific research actually tends to work, incrementally, cautiously, and often in ways that add valuable new layers of understanding without delivering the kind of clean, singular verdict that headlines and video titles are naturally built to promise. The Shroud of Turin remains what it has been for well over a century of serious scientific attention, an object carrying genuine, well-documented mysteries, alongside genuine, well-documented scientific challenges to its traditional identification, with new research continuing to add detail and nuance to that picture without fully resolving it in either direction. This latest DNA study is a real, methodologically serious contribution to that ongoing body of research. It just isn't quite the case, closing the Vatican-shaking revelation that the most dramatic corners of the internet made it out to be, and understanding exactly where the line sits between what was genuinely discovered and what got added afterward through overheated headline writing is precisely the kind of careful thinking this endlessly fascinating 400-year-old mystery keeps demanding from anyone who wants to actually understand it, rather than just react to the latest viral claim about it.